Okay, so today we're going to be talking about acid-base balance and regulation of normal pH within the internal and external compartments within the body of fish. So intracellular pH and extracellular pH, and the different mechanisms that fish use to maintain pH within those physiological optima. Now again, this may involve different pH values in different compartments within the body. So for example, the typical physiological optimum for blood pH or plasma pH is usually between 7.7 and 8.0. The optimum for intracellular pH for most fish is a little bit lower, between 7.2 and 7.5. Now, in the case of the stomach of true fish, the stomach of truly gastric fish, the pH may be as low as 4, whereas the rest of the gut is about 7. So again, what constitutes physiological optima really depend upon the cells or the tissues of the compartment within the body, but a lot of the mechanisms that are used by fish to maintain those different optima are similar. So that's what we're going to be talking about in this lecture. The aquatic environment itself actually does put a few restrictions on acid-base balancing relative to air-breathing vertebrates. The fact that we can simply exhale gaseous CO2 into the environment as terrestrial vertebrates is a major advantage in terms of the ease of acid-base balancing. The fact that fish cannot do that, that they cannot excrete CO2 directly and have to convert it to bicarbonate and hydrogen ions for excretion, really limits what they're able to do in terms of the speed by which they can compensate for acid-base shifts. So acid-base balancing typically involves a multi-tiered process in fishes, and it occurs over different time periods. So the first part, instantaneous buffering or internal buffering, occurs over a period of seconds to minutes. That's why it's referred to as instantaneous buffering. This is the buffering that would occur within that specific compartment. So for example, if aerobic respiration is occurring within a particular cell, it's going to be producing some CO2. That CO2 acts as an acid but can be buffered by the presence of additional bicarbonate ions or organic or inorganic phosphates or other buffers that may be dissolved within the intracellular fluid of that particular cell. So it's a very local, very rapid buffering. Now, the second phase includes respiratory adjustments. These occur over a somewhat longer period of time, maybe minutes, maybe as long as hours, but in most cases just minutes. This is where the fish is going to increase its ventilation rate to either increase or decrease the ability to excrete CO2 that's produced as a result of aerobic respiration at the gill. The third phase is the import or export of acid-base relevant molecules to or from the environment. Now this takes a lot more time. This occurs over a period of minutes to hours, and this is of course related to the respiratory adjustments. When we talk about the transport of pH relevant molecules, we're basically talking about any molecule, any ion that has an effect on pH. So, of course, the most common would, of course, be hydrogen ions themselves. So, as hydrogen ions may accumulate as a result of respiration, the fish may have some instantaneous buffering that goes on immediately to try to compensate for the production of H plus at a particular in a particular cell or at a particular tissue. Then it may undergo some respiratory adjustments, increasing ventilation rate to circulate that blood through the gills to allow for that hydrogen ion to be offloaded. And then there's the matter of actually offloading the hydrogen ions. So what can we exchange ion for ion with the environment to trade? So hydrogen ions being a positive cation, we want to get rid of that and export that to the environment. That can perhaps be linked with import of another positive cation like sodium. So again, each of these processes is going to be occurring, in conjunction. The instantaneous buffering will do what it can, but then respiratory adjustments and exchange of pH relevant molecules at the gill are going to have to come in to complete that compensation to ensure that the buffering system that is available at the cells and at the tissues is regenerated and reconstituted and prepared for the next pH shift. We don't actually have a lot of slides for this next portion of the lecture, so I'm just going to leave this particular slide where it is. We'll pick up with this in a moment, but I want to mention a few things here that, again, we don't have slides for, but are important to understand. We talk about pH shift or acid-base balance that fish need to compensate for, but we need to understand the types of processes that can affect acid-base balance. I've already mentioned one of them, and that's metabolism. Metabolism, specifically aerobic respiration, has a decreasing effect on pH. So this tends to decrease pH over time because metabolism and aerobic respiration produce CO2 as a byproduct. CO2, of course, acts as an acid. It is a weak acid. And so, of course, metabolism has this effect of decreasing pH. Exercise and lactic acid accumulation, of course, have the same effect. Keep in mind, too, that exercise is essentially metabolism at a higher level. So general metabolism has an effect of decreasing pH, so does increased metabolism associated with exercise. That includes, again, both aerobic respiration and anaerobic respiration. In the case of aerobic respiration, it's mostly CO2 that's being produced. That's what's reducing your pH. In the case of anaerobic respiration, it's the lactic acid and CO2 that are both being produced that have the same effect. Some environmental effects that can have an effect on acid-base balance include hypercapnia. Hypercapnia is a term that means you have an excess of CO2 dissolved in the water that the fish is inhabiting. This also has an effect of decreasing pH. It essentially decreases pH in the body of the fish by inhibiting the ability of that fish to excrete CO2 to the environment. The CO2 accumulates in its body, therefore decreasing pH over time. There is one effect that can increase pH, and that is ammonia excretion. Ammonia, NH3, is what's produced by the fish, but it's commonly excreted as a cation, as ionized ammonia, NH4+. So one of the hydrogen ions is being essentially attached to the ammonia molecule, and then they're being excreted together. So this net export of hydrogen ions by coupling hydrogen to ammonia results in an increase in pH. So there are lots of different processes that are occurring within the body of the fish at any time that will have changes that need to be dealt with at a physiological level. So let's talk a little bit about what some of these shifts look like. So we're going to return to the slide at this point. What you're looking at is a figure showing the concentration of bicarbonate ions in the plasma of fish and the pH as a result of that. So along the x-axis you have plasma pH going from about 7.3 to as high as 7.8. Keep in mind normal for most fish is between 7.7 and 7.8. And then you have the concentrations of bicarb or HCO3 minus ions on the y-axis. The point A here, the square, the dark square, that's considered the control point. So again, plasma pH is about 7.75. The concentration of bicarb ions is about 5 millimoles per liter. And in this case, the PCO2, or the pressure of CO2 in millimeters of mercury, is about 2. And you can see that those kind of arched lines going across, those represent different pressures of CO2. So the point A is on the 2 millimeter of mercury line. You can see there's 1, 2, 3, 5, and 7. So again, A represents the control point. Now the shift from A to B is a process that's referred to as metabolic acidosis. This is also referred to as the addition of fixed acid to the system. So for example, if a fish was respiring anaerobically, it would be producing lactic acid. Lactic acid is considered a fixed acid essentially because it doesn't participate in the bicarbonate buffering system the same way that CO2 does. So when we're talking about fixed acids, we're basically talking about anything but carbon dioxide. Okay, so this metabolic acidosis, this is again the result of lactic acid accumulation primarily. This could either be a result of the fish exercising with limited oxygen, or it could be induced experimentally simply by injecting lactic acid into the fish. So we're not seeing an accumulation of CO2. You can see we're just moving down along that 2 millimeter of mercury CO2 gradient. So the abundance of CO2 hasn't changed, but the pH certainly has. The pH has dropped to about 7.45. And as a result of that, the concentration of bicarbonate ions has dropped quite a bit, from about 5 millimoles per liter of plasma to about half that, 2.5 millimoles per liter. So what's happening here is the lactic acid is being buffered by the bicarbonate buffer system, and in doing so, it's consuming some of that bicarbonate ion. So we have less of that available in the body of the fish. As a result of this, at point B, the buffering capacity of the plasma at this point would be reduced. So if there were another reduction, another introduction of fixed acid at point B, the decline in pH after that would then be much more dramatic because there's less buffering capacity in the plasma, simply because some of it's already been used up. Now, in the case of point C and the movement from point A to point C, this is what's considered respiratory acidosis. This is a decrease in pH caused by an increase in CO2 in the system. So you can see that we've moved from the CO2 gradient of 2 millimeters of mercury to the one that's approximately 5 millimeters of mercury. How would this occur? It would simply occur as a result of increased aerobic respiration. So again, this is different from the metabolic acidosis or the addition of fixed acid because in this case we do have an increasing accumulation of CO2. Now, in this case, the pH also declines, although not as much as in the case of the metabolic acidosis, but we do have some shift in pH. The other thing to note here is that in this case we actually have an increase in the concentration of bicarbonate ions. Keep in mind again that the CO2 does participate in the bicarbonate buffer system, and so the increasing availability of CO2 is going to push that bicarbonate buffer equilibrium to form more bicarbonate ions. That's why you have an increase in the concentration of bicarbonate ions here. So in this case, the theoretical buffering capacity of the blood, in this case, or the plasma, would actually be increased somewhat. But again, it's not just the bicarbonate buffer system that participates in buffering capacity. Although we have an increase in bicarbonate ions, in this case you can still see that we had a decrease in plasma pH. There are other what are called non-bicarbonate buffers that are simply not part of the bicarbonate buffer system that can assist in preventing major shifts in plasma pH. And that's actually why you see point C, it has less of a drop in plasma pH than point B. This is because the non-bicarbonate buffer systems are much more effective in dealing with respiratory acidosis than they are in dealing with metabolic acidosis. So as you'll see later on in this lecture, it's actually the non-bicarbonate buffers that tend to have a stronger effect in most compartments of the body in terms of preventing pH change. But certainly the bicarbonate buffer system is involved as well. So again, just a distinction between these two different types of acidosis: metabolic acidosis, which is primarily associated with lactic acid accumulation, and anaerobic respiration, which is more difficult to recover from. And then you have respiratory acidosis, which is just the result of normal respiration, aerobic respiration, which is a little bit less dramatic of an effect and is easier to recover from in most cases. With respect to these non-bicarbonate buffers, I'll note here that these can include things like inorganic and organic phosphates, amino acids, proteins. These are essentially any kind of molecule that can act like a buffer, something that can donate or receive hydrogen ions by participating in these buffer equilibria. One slide. One other thing that I'd like to note about these intracellular values as opposed to extracellular values is that generally speaking, the intracellular pHs for fish tend to be the same as they are in mammals. However, the extracellular fluid and plasma values tend to be much lower. pHs tend to be lower in fish plasma than they do in terrestrial vertebrate plasma, because fish, generally speaking, have lower hematocrits and lower plasma bicarbonate ion concentrations. Generally speaking, the availability of bicarbonate ions in the plasma of fish is very low simply because fish have a very high ventilation rate relative to terrestrial vertebrates. Keep in mind that fish have to move a lot of water across their gills to acquire an appropriate amount of oxygen. Because they're moving so much water across their gills to get that limiting oxygen, um, they lose a lot of CO2 as a result of that. They lose it in the form of bicarbonate ions. So as they're moving all this water across their gill, they're doing so to acquire oxygen, but as a result of that, any CO2 that might be converted to bicarbonate ions and accumulate in the blood is immediately stripped out. Because as soon as it accumulates to a higher level in the bloodstream relative to the environment, it simply diffuses out or may actually be exchanged. And so again, this solubility of CO2 and bicarbonate ions in water is going to drive most of that out. Now, in the case of terrestrial animals, we don't lose bicarbonate ions as a result of respiration. We can't lose them to air as a respiratory medium the way that fish can lose bicarbonate ions to an aquatic to water as their respiratory medium. And so simply as a result of that, the loss of those bicarbonate ions to the environment, fish can't accumulate as much of those bicarbonate ions, which results in their bicarbonate ion concentrations and plasma pH levels tending to be lower than they are in terrestrial animals. So this table summarizes a whole lot of different information here about the types of situations fish might find themselves in and what kinds of acid-base changes that can induce. I'm just going to highlight a couple of them. We're going to begin with hypercapnia in the water. So hypercapnia, again, is an accumulation of CO2 in the water that the fish is living in. This increase in environmental CO2 is actually going to prevent and slow down any diffusion of CO2 that might occur at the gill. This results in an increase in CO2 in the blood. Now, how could this happen? You can induce high levels of CO2 in the environment experimentally, but keep in mind this can occur naturally as well. If you have water that is poorly mixed, that's covered by vegetation, etc., this can lead to high levels of CO2 in the environment. Okay, so we have high levels of CO2 in the environment, either because of natural or induced causes. That interferes with the fish's ability to release CO2 at the gill. We talked about that when we talked about the respiration system of fishes in that lecture. And then that creates these shifts in pH within the body. So you can see here the very first row, water hypercapnia results in internal hypercapnia, or an accumulation of CO2 in the body. That increasing CO2 availability has the effect of reducing pH. So the compensatory response in the extracellular fluid is to increase the plasma bicarbonate ion concentrations. That's exactly what we saw in the figure at the beginning of the lecture when we moved from point A to point C. Now, the degree of compensation here is relatively good, and ultimately what the fish will do is they will export hydrogen ions at the gill as a part of a respiratory adjustment to deal with that pH change. Now, recovery here in this case is actually going to take place over a period of days, and that's only for an internal pH shift of maybe a tenth to 2/10 of a pH unit. I really want to stress how challenging acid-base compensation, maintenance of acid-base balance is in fish. It takes a good deal of energy and it takes a good deal of time in order to compensate for these shifts that can occur. This is also part of the reason that I think it's very unwise to use CO2 as a sedative in fish. I'm sure many people that are listening to this lecture have used carbon dioxide as a sedative. You can certainly knock out fish fairly easily with it, but what you're essentially doing is creating a very high degree of water hypercapnia. Of course, that will result in hypercapnia in the fish, and as you can see, it causes this pH shift that fish can compensate for and can recover from, but it's going to take them days to recover from even a relatively minor internal acid-base shift. So usually when you're anesthetizing fish in the field with CO2 or even in the lab, you want those fish to recover quickly, and you want them to go back into their environment and be functional. You're definitely putting fish at a major disadvantage, both bioenergetically in terms of basic physiological function, by inducing hypercapnia and using that as your primary means of sedation. So really not a good idea to be using CO2 as a sedative for a whole host of different reasons. Interestingly, hypercapnia, or excuse me, hyperoxia in the environment has a very similar effect. So water hyperoxia, that's the second row here, this is extremely high levels of oxygen in the environment. It causes hypercapnia and all of the same types of responses in fish, simply because the increasing concentration of oxygen in the water is going to slow down the fish's ventilation rate. They simply don't have to move as much water across the gill to acquire oxygen when oxygen is really, really high. So they slow down their ventilation rate because they don't need to have a high ventilation rate to acquire oxygen. But as a result of that, their ability to lose CO2 and offload CO2 to the environment is reduced. And so that some of that CO2 begins to accumulate in their plasma. So both water hypercapnia and water hyperoxia have the same effect, and that both can induce hypercapnia. So I've already talked about why it's not advisable to use carbon dioxide as a sedative because it causes hypercapnia. It's also not advisable to maintain extremely high levels of oxygen in the water. Even though you might think that you're doing a good thing by providing the fish with excess levels of oxygen, you may be inducing hypercapnia and, again, some of the same problems that would be associated with water hypercapnia. You can again see a variety of different other factors here that are described. Metabolic acidosis can be created either through exercise or through an injection of lactic acid that you can do experimentally. So you can see that, let's see, towards the bottom here, acid infusion or base infusion. We're going to deal with acid infusion. This is, again, can be induced experimentally, but it can also occur during normal anaerobic respiration. As a result of that, again, you see metabolic acidosis resulting here. Again, it's not hypercapnia because this isn't the result of CO2 accumulation. It's the result of lactic acid accumulation. Of course, you see a pH decrease and some of the same effects that we saw illustrated on that figure at the very beginning of the lecture. Another thing that can be a physiological adjustment to pH imbalance, one thing that can facilitate that response rather, is salinity. So generally speaking, when we're trying to compensate for some sort of acid-base imbalance, it's going to involve that transport of acid-base relevant molecules at the gill. So we're going to be exporting or importing bicarbonate ions, hydrogen ions, etc. That process can be linked with sodium or chloride transport. And so if more of those ions are available, that can actually facilitate the return to normal acid-base conditions. That's what this figure is showing you. So you have time along the x-axis, and this is essentially the time of exposure of fish to different types of different dilutions of salinity. So you can see the very top line, it's a dashed line with triangles. The SW here refers to full-strength seawater. Then you can see moving down the slide 20%, 8%, and 4%. These are 28 and 4% dilutions of full-strength seawater. So 20% of full-strength seawater, 8%, 4% of full-strength seawater. So these fish were exposed to these different salinities. These are a euryhaline fish, so they can osmoregulate normally in each of these salinities, but the curves look different, and that's because the availability of sodium and chloride in the higher salinity solutions can facilitate acid-base balance, or rather acid-base balancing in this species. The y-axis here actually represents the net efflux of hydrogen ions. So this is how many hydrogen ions these fish are able to export to the environment. So you can see that we have the zero line. This would mean that there are no, there's essentially no net exchange. So this doesn't mean that the fish aren't exporting some hydrogen ions. They are. They're just not exporting as many as they're exporting the exact same number that they are producing in terms of normal respiration. So for the fish that are in full strength seawater, the one at the top, you can see that has the highest level of efflux. So those fish are exporting a greater number of hydrogen ions than the fish that are held at 20%, 8%, or certainly from 4%. The 4% fish are actually, they have a net influx of hydrogen ions. You can see they have a negative efflux. So they are not exporting as many hydrogen ions as they should be. They're producing more through normal respiration and normal metabolism than they are able to export. Again, the rationale for why you have greater hydrogen ion exports in these higher salinities is because the export process is linked to the import of needed sodium and chloride. So again, if this is a freshwater fish, we're talking about a very adaptive process. We're producing excess hydrogen ions and excess bicarbonate ions as a result of the shift of that bicarbonate buffer system in response to CO2 production. So we're producing more of those. We have excess hydrogen ions and bicarb ions. And as a freshwater fish, we need to be continuously importing sodium and chloride ions. So if we can link those two processes and export a hydrogen and import a sodium, and likewise export a bicarbonate ion and import a chloride ion, this is going to be a really beneficial thing for the fish. It's going to be very advantageous energetically. If there are more sodium and chloride ions available in the environment, it's going to make that process easier. And that's, again, what this figure is illustrating. The fish that are in the 8, 20, and full-strength seawater solutions are able to acquire sodium and chloride more easily at the gill because they're more abundant in the water, and so they can exchange more hydrogen ions and actually more bicarb ions as well in exchange for those sodiums and chlorides. Now, in the case of the fish held at 4%, they would very much like to export some of those hydrogen ions that are accumulating, but they're having a tough time finding sodium ions to exchange them for. And so because they're not able to find, so to speak, those sodium ions, they're not able to make those exchanges at the gill, and so their net export is not keeping up with their production of hydrogen ions in the body, and so that's why you see this negative hydrogen efflux. It actually represents an accumulation of hydrogen ions in the system. So again, this is a very adaptive process in the case of freshwater fish. Saltwater fish, they're actually able to export hydrogen ions and bicarb ions much more rapidly because there's an abundance of sodium and chloride in the environment to exchange with. That's exactly what you see here in terms of the saltwater solution fish. They export that hydrogen very easily because there's plenty of sodium to trade. But again, in a saltwater fish, the last thing that they want to do is import more sodium and chloride. However, that very process does occur in saltwater fish. Saltwater fish do export and exchange hydrogen and bicarb ions going out for sodium and chloride ions coming in, even though that's counter to what they would want to do from an osmoregulatory perspective. This should really drive home the point that acid-base balance, although we might not think about it as often in fish as we think about osmoregulation, the importance of acid-base balance will trump osmoregulation every time. From a physiological basis, if you're trying to maintain normal physiological functioning, either intracellularly or extracellularly, it's much more important to maintain pH ranges than it is to maintain osmolality ranges. So although the saltwater adapted fish is going to be acquiring more sodium and chloride than it wants, it will do that to get rid of the hydrogen and the bicarb ions that it needs to in order to maintain its pH. It will have to deal with those additional sodium and chloride ions that it has acquired and now needs to get rid of, but it will do that and it will expend that energy in order to excrete sodium and chloride. It will do that as long as it's able to maintain its pH first. So again, the fact that these exchanges happen in saltwater fish the same way that they happen in freshwater fish tells you how important These are just a few figures to illustrate exactly how that happens. So the first slides that we're dealing with here, this is for a freshwater adapted fish. On the left-hand panel, you have control conditions, and then on the right-hand side, you have acidosis conditions. So keep in mind that this could be any type of acidosis. This could be either metabolic or respiratory acidosis occurring in the fish because of either increased anaerobic respiration or increased aerobic respiration. It could also be acidosis induced by hypercapnia in the environment, or any number of the other types of acid-base balance effects that were summarized on that previous table. For whatever reason, the fish in the right-hand side is undergoing acidosis, and the cells of the gill are going to compensate as a result of that. So on both panels, you can see the water is on the left-hand side. The cells that are shown, those are the cells of the gill epithelium, and then you have the extracellular fluid on the right-hand side. So extracellular fluid in this case is simply the blood or the plasma of the fish. So you can see the pavement cells highlighted here as PVC, and then you can see the chloride cells, the mitochondrial rich cells, shown in the middle. Now, under normal conditions, you can see that there are those hydrogen ion ATPase sodium pumps that are present in the pavement cells, and perhaps to a lesser extent the chloride cells as well. These are trading hydrogen ions for sodium ions under normal conditions. So this is something that we discussed, the pavement cells being involved in when we talked about osmoregulation earlier in the course. So those pavement cells are going to be actively exporting hydrogen, and that's going to encourage sodium ion diffusion because of the charge imbalance that export of hydrogen ions is going to create. Now, I should note here that for this slide and for the next slide as well, any of the circles that are shown in dark color, or the filled in or closed circles, those are transporters that require ATP. The open circles are simply transporters that are exchanging that do not require ATP, so they don't require energy to function. And then dashed lines on these figures represent simple diffusion. So under normal conditions, again, on the left-hand side, you have CO2 being produced through the actions of carbonic anhydrase. That's going to be converted to hydrogen ions and bicarbonate ions, and the hydrogen ions are going to be then actively exported to the environment using those hydrogen-sodium ATPase pumps. Now, in the case of the freshwater fish under acidosis conditions, you can see that the number of those hydrogen sodium pumps has increased, whereas before we had only 2 of them functioning. Now we have 4 of them functioning. So during those acidosis conditions, we're going to ramp up the number and functionality of those pumps so that we can push out more hydrogen ions. And of course, then we're going to be acquiring more sodium as a result of that, which is a positive thing. The other thing that's going to be occurring here is that CO2, bicarbonate ion transport in exchange for chloride is going to be a major means of eliminating excess CO2. So the CO2 is converted to hydrogen and bicarbonate ions. We just talked about how bicarbonate ions are going to be pumped out. The bicarbonate ions can be exchanged for chloride. So you see that under normal conditions. We have this co-transporter that is getting rid of bicarbonate ions and bringing in the negative chloride ion in exchange. Again, this is a very positive thing. However, keep in mind that the presence of bicarbonate ions is part of what contributes to that extracellular fluid buffering capacity. And so we don't want to lose all of the bicarbonate ions that we have. So under acidosis conditions, you can see that not only is there a change in the number of hydrogen ion transporters, there's also a morphological change. Under normal conditions, the chloride cells in these freshwater fish have lots of surface area. We talked about why that is during a previous lecture. But during acidosis, the chloride cell actually becomes closed off. The pavement cells cover it up to try to prevent too much of that bicarbonate ion loss. Again, we're trying to retain that buffering capacity, limited though it is, of the extracellular fluid. So what we want to do is encourage excess export or increased export of hydrogen ions out, and we want to limit the loss of bicarbonate ions at the same time. And so that's what you can see here on this right-hand panel. Again, we now have double the number of hydrogen exporting pumps that are going to be pushing out hydrogen ions and therefore attracting some additional sodium to flow in. But because the pavement cells are covering up the chloride cell is somewhat retracted, we're going to be limiting the amount, limiting the loss of bicarbonate ions in exchange for chloride ions simply by restricting surface area that's available. And you can actually see this in the micrographs of the gill structure. So in the upper panel, this is a fish under normal conditions, freshwater fish under normal conditions. So you can see those broad patches that represent the surface of the chloride cell. That's in what's highlighted with the arrows there in the B panel. The A panel actually shows a zoomed in portion of the arrow. So that's all those little finger-like projections increasing the surface area of that chloride cell further. So A and B, that's under normal conditions. C and D is the same fish, the same freshwater fish under conditions of acidosis. In this case, this was acidosis induced by 6 hours of environmental hypercapnia. So they expose these bullheads to high levels of environmental CO2 for a period of 6 hours, and then you can see the changes that have occurred at the gill. And now, instead of having these broad patches with all these little finger-like projections, now we have closed off little pores. So you can see a few of them are highlighted with the arrows in panel C. In panel D, you can see the zoomed-in version of it. Now, instead of looking like a normal freshwater adapted chloride cell, it looks a lot more like a saltwater adapted chloride cell. So instead of having this broad patch with finger-like projections, now we have just these small closed-off pits. So simply by restricting surface area, we can decrease the loss of bicarbonate ion. We can decrease the loss of bicarbonate ions to the environment, while at the same time still increasing the export of hydrogen ions to the environment by the surrounding pavement cells. Now we have another figure here showing essentially the exact same processes occurring for acid-base balance in seawater adapted fish. So you can see that we still have the hydrogen sodium pumps that are active, and we are exchanging hydrogen ions for sodium ions. Now in this case, these are the open co-transporters of the chloride cell. It may be that the seawater adapted pavement cells also engage in the same active ATP using hydrogen export as well. But generally speaking, there is less of a need in seawater to actively use those hydrogen, the ATP-driven hydrogen pumps of the pavement cells, simply because these fish don't have to work that hard to find sodium ions to exchange hydrogen for. So that's why you see in this case open circles indicating the hydrogen-sodium co-transporters functioning in the chloride cells, unlike the emphasis on the active ATP-utilizing hydrogen pumps that we saw in the freshwater fish. Now, of course, what you see under conditions of acidosis is an increase in the number of those co-transporters. So these don't utilize ATP, but the number of those co-transporters will increase in seawater fish exposed to acidosis, again, to encourage that hydrogen ion export. You'll also see the same kind of thing, the same rapid increase in bicarbonate ion co-transporters, more of that bicarbonate being pushed out. Again, keep in mind that this process of acid-base balancing and hydrogen ion export is counter to what these fish want in terms of osmoregulation. So they're getting rid of their hydrogen and they're getting rid of their bicarbonate ions that accumulate as a result of CO2 production, but they're acquiring additional sodium chloride as a result of that. They will engage in these acid-base balancing efforts that are counter to osmoregulatory efforts because acid-base shifts will have a greater negative effect on the ability of the fish to function physiologically than the effects of a bit of osmoregulatory shift.